There's a mystery written deep inside all of our DNA, a real genetic whodunit that separates us from our closest animal relatives. And today, we're going to crack the case. So, it all starts with this one simple question that's actually kind of profound. If we're so closely related to the great apes, how come we have 46 chromosomes, but they all have 48? Where did two of ours go? Did they just poof, vanish? And that's what makes this whole thing so baffling. I mean, we share about 99% of our DNA with chimpanzees. Our genetic scripts are practically identical, and yet they're packaged into a different number of volumes. So what on earth happened? All right, to figure this out, we've got to put on our detective hats and follow the genetic breadcrumbs back in time. Okay, so the simplest explanation is that two entire chromosomes just got lost somewhere along our evolutionary journey. But here's the thing. In genetics, losing a whole instruction manual is, well, it's usually catastrophic. So something else has to be going on here.
Now, before we can spot the clues, we need to understand the crime scene. So let's take a quick look at the structure of a chromosome itself.
Just imagine your entire genome is this massive library of instructions, right? Each chromosome is one book in that library, totally packed with genes that tell your body how to build itself and what to do. And every single chromosome has a pretty distinct anatomy. For our investigation, there are two parts that are absolutely crucial. The centromere, that's the pinched-in waist part, and the telomeres, which are the little protective cabs at the very ends. These are the genetic fingerprints we're going to be looking for. The best way to think about a telomere, it's just like that little plastic tip on your shoelaces. Its job is to stop the chromosome ends from unraveling. But, and listen up, this is key, they also stop them from getting sticky and attaching to other chromosomes. Okay, so armed with that knowledge, genetic detectives can actually compare chromosomes between species, looking for these telltale signs of ancient events that completely reshaped our genome.
See, chromosomes aren't set in stone. Over millions of years, they can break, they can merge, they can even flip upside down. The main suspects in our case are either a fusion, where two chromosomes stick together, or a fission, where one splits apart. Since we went from 48 down to 46, all signs are pointing to a fusion event.
So how do we prove it? Well, scientists use this really cool staining technique that makes unique light and dark bands appear on each chromosome. It basically creates a genetic barcode. So the mission was simple. Find a human chromosome whose barcode is a perfect end-to-end match for two separate ape chromosomes. You find that, and you've found your culprit.
And that barcode comparison, it leads us directly to the star piece of evidence, the absolute smoking gun in this entire genetic mystery.
The theory sounds great, right? A fusion must have happened. But a theory is nothing without proof. So where is that smoking gun? It's right there, inside pretty much every single cell in your body. The evidence isn't missing at all. It's been embedded within human chromosome 2 this whole time.
The scientific conclusion is just undeniable. At some point deep in our ancestry, two chromosomes that are still separate in other great apes fused together, tip to tip. And that created the single, massive chromosome we now call our chromosome 2.
And let me tell you, the proof is overwhelming. First, the barcode. The banding pattern on our chromosome 2 lines up perfectly with two smaller separate chromosomes in chimps, gorillas, and orangutans. Check. Second, it has a ghost of its past, a second inactive centromere, just sitting there right where you'd expect it to be if two chromosomes had merged. And the final nail in the coffin? Right in the middle, at the fusion site, we find leftover telomere DNA. Those sticky ends that got joined together, it's literally a genetic fossil. So there you have it. We never lost any chromosomes at all. We just reorganized the library. Two smaller books were bound together into one larger volume. Case closed. But solving the case is really just the beginning. Why does any of this even matter? What does this grand reshuffling of our genes actually tell us about who we are? Well, as you can see here, that massive fusion event was definitely the most dramatic change, but it wasn't the only one. Our genome also has at least 9 major inversions. That's where whole sections of chromosomes got flipped around, plus a bunch of other tweaks that separate our genetic blueprint from a chimp's. And here's why that is so important. When you shuffle your genetic deck that dramatically, it can create something called a reproductive barrier. Think about it, an ancestor with 46 chromosomes trying to have a baby with one that has 48. It would be a problem. Their chromosomes wouldn't pair up correctly to make viable offspring. This genetic incompatibility is one of the key engines of speciation. It's what allowed our ancestors to split off and forge their very own evolutionary path.
And this fusion isn't the whole story. Scientists have found other hotspots of superfast evolution, like this one area on the Y chromosome called AZFA, which is critical for male fertility. It's even more proof that our genome has been actively changing in ways that directly impact our biology and define what makes our species unique.
So, you see, this ancient fusion event wasn't just some quirky accident. It was a pivotal chapter in the story of what made us human. It reminds us that our genome isn't a static, dusty blueprint, but a dynamic, living document. Which leaves us all with one final question to ponder. What new chapters is it writing today?